a pleasure to be here and have the opportunity to speak to you. So those who attended the talk the day before yesterday, this will be totally different. No physics, not much theory. This is my applied side, if you wish. So as Laurent already mentioned, I work in the Bernoulli Institute, which used to be the Johann Bernoulli Institute until recently, but we got rid of the Johann in order to confuse people a bit more. So they don't know which one we are referring to. But Johann Bernoulli actually worked in Groningen, not in this building yet, but he worked for quite a few years in Groningen a long time ago. Okay, so the talk will be about prototype-based classifiers in combination with what we call relevance learning, or sometimes it's called metric learning. There are different names for the same thing. And I want to illustrate and discuss this in the context of medical applications, biomedical applications. So I will first briefly introduce you to prototype and distance-based systems. And the working horse, my favorite working horse in this context, is the so-called learning vector quantization, which goes back to Groningen already, so it's already quite old as a concept. And it can be extended by adaptive distance measures, or relevance learning as we call it, to make it even more powerful and more flexible. And I will illustrate these ideas in terms of three biomedical application examples. One is about the tumor classification in tumors of the adrenal gland. I will tell you a bit more about that. There, the classification is based on metabolomics data, steroids to be more precise. Then the second example relates to rheumatoid arthritis and its diagnosis in very early stages of the disease, where the data corresponds to cytokine expression in synovial tissue. And in the third, if I have the time, the third application example starts from 3D images, brain scans, PET scans of brains, and the goal is to diagnose and discriminate different neurodegenerative diseases like Parkinson's and Alzheimer. But we don't work with the images themselves, we work with a low-dimensional representation of the images. And then if time allows, I summarize and briefly discuss a few general challenges in the context of biomedical data. So these are the three application examples that I will go through, but first let me talk a little bit about the classification framework that we use, the tools that we use. So in supervised learning, generally speaking, which could be a classification, a regression, or prediction in a time series, something like that, we want to train a system based on labeled example data. And the generic workflow is, of course, that we have some example data. We train a model or a network or an LBQ system, whatever you're using. And then later in the working phase, we want to apply it to novel data. And hopefully it performs well and does what we trained it to do. And usually, in order to have an idea of how well our system will do in the novel data, with respect to novel data, we insert here a loop that you might want to call validation, think of cross-validation, similar schemes in which we can estimate the performance that we can expect for novel data. So far, so good. So in supervised learning, the big advantage compared to unsupervised learning is that we at least have a feeling, or we know what the goal of the exercise is. So there are more or less obvious performance measures which we can use to evaluate the performance of our model on test data and also maybe later on real world data. So we can look at accuracies or errors, to put it in a negative way. Overall class-wise accuracies, we can look into greater detail into things like the precision recall characteristics or the receiver operating characteristics. So in supervised learning, we know what the goal is. That makes life a bit simpler than in other settings where this may not be so very clear. Now, I will focus on learning vector quantization, which is a system or a framework that works with prototypes and distances. And distance-based classification is the almost, well, in a sense, the most obvious idea that you could have for classification. And probably everybody is familiar with the nearest neighbor classifier or the k-nearest neighbor classifier as a concept. What do we do there? Well, we simply store a set of labeled examples, observations that we made previously in history. And if we have, like here in this sketch here, two-dimensional feature space, then any of these points might be an earlier observation. And we also know the class membership of these observations. So we have the observations that belong to the blue class, to the red, and to the green. And we simply store those. And now if we have a new observation, in medical terms a new patient for instance, we look up in our history of observations which of these data points is closest to the new point. And then we let the nearest neighbor in our data set decide, or if we want to make it a little bit more fancy, we look at the 5 nearest neighbors and have a majority vote, something along these lines. But we're just comparing to the cases that we have seen in history. Now, locally, this obviously, if we think of measuring similarity or distance in terms of Euclidean distance here, that gives me a little piece of a linear decision boundary that here discriminates between the green and the blue. And if I go through my data set, then I come up with something like this, which is a very wiggly decision boundary discriminating these 3 classes, right? Now, this is awfully simple as a concept, so that's a big advantage. There's no training phase whatsoever. All you do essentially is store your data points, right? So you're not working hard adapting some model or something. You're just storing your data before you start classifying. And essentially, there's only one parameter, which could be the k in the k nearest neighbor, for instance. And of course, you have to make a choice. What is the distance function? What is the distance measure that you use for comparing observations? That's a crucial point. Now, this can get, these are the downsides, this can get expensive, not only because of the storage, but mainly because of the computation. In principle, for a new data point, you would have to compute all distances to all the data that you've seen before, and that can get expensive. Then find the minimum or the k minimal distances, which also can constitute considerable computational effort. And your system will be sensitive to mislabeled data or outliers. So here, in this case, for instance, we would probably not trust this point to be really a member of the blue class, but our nearest neighbor classifier would define here a bubble, a region, an island, in which any new observation will be assigned to the blue class, right? So we get in tendency an overly complex decision boundary from this idea very often in practice. Okay, so this is exactly the idea, or learning vector quantization has exactly the aim to overcome these difficulties, at least to a certain extent. And the idea is to replace our data, instead of using all these individual data points, to replace our observations by something that we might want to call typical examples of the classes. They're called prototypes. So we represent the data by several prototypes. There's a typical, presumably typical green case, a typical observation in the red class, and for the fun of it, I've used here 2 prototypes for the blue class. And now I can do, similar to the nearest neighbor classification, I can now do nearest prototype classification. So if this is my new observation, I compare it only with the prototypes that I'm storing, not with the individual data points anymore. And here I would say, oh, the nearest prototype is the green one, so I assign that data point to the green class. And that gives you, obviously, if we stick with Euclidean distance, that gives you much smoother, much simpler decision boundaries between the classes, and it makes you less sensitive to single point labels, right? Okay. Now, this is, in that sense, very robust, much more robust against outliers, single data points. You have much lower storage needs. Once you've determined the prototypes, you forget about all these individual data points. Computational effort is significantly lower because you only compute distances to a handful of prototypes, typically not a huge data set that you're storing. And compared to other classification schemes, it's a built-in natural choice for multi-class problems. Simply, you need at least one prototype per class, and there you go. Your scheme realizes a multi-class classification. Main advantage from my point of view is that we are parameterizing the classifier still in the same space in which we observe the data. So the prototypes are defined in the feature space, right? So if your data is age and shoe size, also the prototypes can be interpreted as instances of age, and shoe size. If your data is blood pressure profiles over 24 hours, the prototypes can be interpreted in that way. So if you have an LVQ system derived from a data set, you can talk to the domain expert, say the medical doctor, in very plain terms. You can say, look at this. Does this look like a typical case for disease A or disease B? So the LVQ system, the prototypes, are quite intuitive, quite easy to interpret, and that facilitates discussions with the domain experts. So in modern terms, they are white box systems because you really understand what they are doing. There are downsides, obviously, as always. We have to do some sort of model selection here. We have to decide how many prototypes do we want to use per class, what is a good choice for this model parameter, or should we try different settings and then compare their performance in a validation scheme, things like that. And of course, compared to the nearest neighbor, now this requires training. We don't get the prototypes for free. We only have the data, and then we have to decide where to put the prototypes, and that's exactly the purpose of the training process. Now, Kohonen suggested in the first variants of this learning vector quantization also an intuitive, heuristic kind of training prescriptor, and it's plain, simple, and also intuitive. So what we will do is naively initialize prototypes somewhere in our feature space, randomly, which is not a very good idea, but we should at least initialize them where there is some data, but in principle, we can put them anywhere. Now we start presenting example from our back of data. So we present here a single feature vector, which was labeled by the experts as a blue class member. We determine among all prototypes currently the one that is closest according to our distance function, and I'm still using Euclidean distance here. So this would be the closest prototype, which we call the winner. It's supposed to represent blue data, so Kohonen tells us, well, move it even a little bit closer to the observation that you're making. Next data point comes, it's here in our feature space. Experts tell us it belongs to the green class. The nearest prototype in our current configuration would be this guy, but that one is supposed to represent red data, a different class. So Kohonen says, well, push it away from the observation. Now this is essentially heuristics. Well, it makes a lot of sense because what we expect is that in the end, our blue prototypes will go to where we have seen blue data very often, and the red prototype will go where we see red data very often, and in contrast, they will be pushed away from the other classes. So what we hope for is that in the end, we kind of represent the arrangement of classes in feature space. Our prototypes then, together with the distance measure, realize a tessellation of the feature space, which is piecewise linear in the simplest case. And the aim is, of course, to discriminate classes and to generalize for novel data. We want to get the correct answer from the closest prototype in many cases, in as many cases as possible. Okay. Now this LVQ1, as it is called, is the basic algorithm, and the first one suggested by Kohonen. There you find a zoo of variants, more or less heuristic or more or less well founded mathematically in the literature. One particularly attractive one is called Generalized learning vector quantization, which is a stupid name, admittedly, but that's what that people use in the literature. It's generalized in the sense that now it's a cost function based system, and we can define a cost function which can be written as a sum over all examples in our data set, and a term lowercase e associated with each example, where this e is given by this expression. And what do we have here? Well, in here we determine first the closest prototype from the correct class. So if we have a red data, we determine the closest red prototype. And we also determine the closest wrong prototype, which is the closest among all prototypes that would assign our data point to a different class. Now we compute the distance from the closest correct prototype and the distance from the closest wrong prototype, and essentially we want to minimize this quantity while we want to maximize that quantity. So we want to minimize the whole thing here upstairs. This is essentially a scaling or normalization that doesn't worry us much. So if phi here is a monotonically increasing function, we want to minimize all these terms as much as we can. And actually, if the argument is negative, that means that the closest prototype, the actually closest prototype is correct. So the LVQ system would give us the correct answer to that example. So we want to have as many as possible negative here. Now we can play with that function phi, and that has been done extensively in the literature. I will restrict myself to very simple cases only. But for instance, phi could be a sigmoid function that looks like this thing here. And if we make this very steep, essentially we are counting how many terms are negative, how many are positive. So we are counting the number of errors that we're doing on the training data. Plausible. But we can even use a linear function or the identity function and still keep that flavor of in tendency trying to minimize the number of errors, but not explicitly doing it, right? Okay, so the aim will be to classify as many training examples correctly as possible, and we do that by hopefully placing the prototypes in class-typical positions. So far, so good. So this is a cost function-based approach, and people consider it more objective than the heuristic one, but then one of my favorite quotes in the area, in the field, is that there's nothing objective about objective functions, because of course we have made a choice here, and that is guided by some insights, some heuristics ideas. From now on, it's mathematically well-defined, and the mathematicians are happy they can consider convergence properties and the like, but qualitatively the difference is not that big. It's a nice cost function-based approach that we can use and will use a lot. How do we train such a system? Well, we optimize the cost function with respect to the prototype positions in the feature space, and we could, for instance, do stochastic gradient descent as just one example. So for each example, we would determine the currently closest correct prototype and the currently closest wrong prototype and change their position slightly according to the negative gradient with respect to the contribution of the example that I'm looking at. So this would be a minimization of capital E along the ideas of stochastic gradient descent. Just one procedure that we can apply. The nice thing about the GLVQ cost function is that we can plug in any distance function that makes sense and that we can take derivatives of. So we're not bound to use Euclidean distance. There's a question. Oh, the question is, do I need to add regularization to keep the system from instabilities or so? Might happen. GLVQ turns out to be rather robust, but you have to take, there are, of course, terms that I can simply add to the cost function to regularize. I'm only talking about the simplest basic versions of that algorithm. But you're right, this is possible and sometimes necessary. The nice thing is we can insert here a distance function that we believe makes sense. As long as it's differentiable, we can construct the gradients and move on along the lines of generalized learning vector quantization. So if you actually work it out for Euclidean, if you just work it out computing the prefactors, then here you would get in the changes of the prototype positions the derivative of the distance from the closest correct prototype and from the closest wrong prototype. Okay. And we can insert, obviously, whatever we want. If we use Euclidean distance, then the form is rather simple. We have these abbreviations of the prefactors. Then what we're doing is, similar to LVQ1, we're pushing the correct prototypes closer to the data. We're moving the wrong prototypes away from the observation. So we're keeping the same basic idea, the basic flavor of LVQ1, even in GLVQ. Okay. Now, what kind of distance measures can we use? Well, as physicists, we have many at hand. We could go to the Minkowski measures, generalize away from Euclidean distance, which would be the case p equal 2. Knowing something about kernel machines, maybe we want to use kernelized distances where we use a kernel function which is represents a dot product in some abstract higher dimensional feature space, for instance. But then we can construct something that looks like a distance and is based on the kernel. We can use, if our data corresponds to histograms, We can use statistical measures like Kullback-Leibler divergence, which is a bit tricky in practice, but other divergences can be used to compare histograms, for instance, the Cauchy-Schwarz divergence, which I just here show as one example. So there's a zoo of possible differentiable distance measures that we can use, and one possible workflow would be select several according to your insight into the problem, according to some prior knowledge that you have the suspicion this could be a good set of distance measures, or determine them in a data-driven pre-processing step in an unsupervised kind of learning, and then compare the performance in different LBQ systems that use different versions of the distance, for instance, by means of cross-validation. That's one legitimate way to go, but it can be, of course, a lot of work, and you might be missing important choices here. Okay. The question whether we're using the right or the wrong distance really is crucial, obviously. And as an illustration in my lectures, I always discuss the difference between astronomy and astrology as a case of using the wrong distance measure. So if we look at the stars in the sky, and we're using angle instead of physical distance, 3D distance, as to associate stars together, then we might see some structures, like the Big Dipper, which are not structures at all. They're artifacts of using the wrong distance. You can say I'm using angle instead of 3D distance, and that makes me think this is a structure in the sky, or I'm normalizing implicitly to the unit sphere, which is the same, saying the same thing in a different way, right? If you combine this with over-interpreting your data, which we call overfitting, then you might even end up with something totally stupid like this. So using the wrong distance measure can give you totally wrong results, and in terms of a classifier, very bad performance, for instance. Okay, now there's an elegant framework which, at least to a certain degree, circumvents these difficulties. The idea is to use a parameterized distance measure, a family of distances, which can be specified further by setting certain parameters. And then the idea is to optimize the parameters in the same training process in which we push around the prototypes in the training. Okay, so that is called relevance learning, or adaptive distances, or metric learning, depending on context and scientific community. And the one example I want to discuss, which I will use in the practical applications, is called matrix relevance learning vector quantization, in which we optimize the prototypes in the training process, but in the same training, in the same optimization process, we optimize a so-called relevance matrix, which defines a pseudo-Euclidean distance function, as we will see. And this is, I want to point out, this is different from pre-processing. We do both in one go in the same training process. And the simple choice that takes you away a little bit from plain vanilla Euclidean distance would be a quadratic form of this form with a matrix lambda that replaces the identity. For the identity, you would get back the squared Euclidean distance. With the matrix lambda, you can play with the influence of the single features and the combinations of two features with respect to the distance. Now, a crucial thing about distances that we should preserve, I wouldn't even insist on having a metric. For the LVQ, that's not so important. But one thing I would want to keep is that the distance is non-negative. That makes life easy and is convenient. And a simple way to make this non-negative is to say, well, let's reparameterize lambda as some other matrix omega transposed omega. So it's a square. The whole thing becomes a square, and the square is never negative. And now I can just as well say, okay, I parameterize my distance in terms of omega, but this is the distance that I'm using. Okay. And the training then would move around W in feature space, and it will change the entries in the matrix lambda, or omega rather, because that's what I want to work with, right? And how can I do that? Well, I have a cost function based approach. The cost function, so far we interpreted it as a function of the prototype positions, but now we can also interpret it as a function of prototype position and distance parameters, omega. So we can do, for instance, stochastic gradient descent in the same cost function, but now with respect to the omega elements and with respect to the prototypes. That would be one way to go, or any other optimization, nonlinear optimization scheme you want to use. Now we can play with this basic idea. We can have, in the simplest case, like here, one global distance measure for the entire data. Or we have several local distance measures. We can even go as far as saying every prototype has its own distance metric, uses its own matrix to determine distances. We can simplify life. We can say, okay, let's stick with the diagonal matrix, which gets us, again, closer to Euclidean distance, but now we're at least weighting the different features differently. And we can even say, no, well, nobody forces us to use omega as an n by n matrix. We can define it n by m, and that would mean intrinsically I'm mapping, for instance, high-dimensional data to a lower m-dimensional space. I compute Euclidean distance in that space, and then I have a distance defined in the high-dimensional space again. So I can use this for low-dimensional representation of the data and also discriminatory visualization of data sets. I would stick with the simple version where we have one global matrix only. There's a question.
So using such a parameterization usually introduces some non-convexity, the problem, additional non-convexity, which might not be here in the first place if you use lambda. Have you found any problems with this?
Well, it's not the question is, does this impose an additional non-convexity that causes problems in practice? Well, the thing starts off non-convex anyway. The cost function is already non-convex, and in my experience, there's no proof. It doesn't add difficulty to the thing. Another difficulty that you could point at this point in time is another difficulty that you might expect is because you're introducing a whole lot of additional parameters to the system, you might expect overfitting problems, right? Because you're making your space of free parameters much larger. So the thing could be prone to overfitting, but it's not in practice in most cases because the matrix omega in the end of the training process, we first observed that empirically and then we can show that it does systematically, becomes a very low rank matrix. So the effective number of degrees of freedom is actually still linear in the dimension of the data, but that's only a side remark. Okay, so what do we get from that? If it all works, if this weird idea works, after training we get the prototypes. They should represent typical class properties of the data, right? Or subtypes if we use several prototypes within one class. But we also get the relevance matrix. And we can read off information from the relevance matrix. We can interpret it, at least in a heuristic way. The elements lambda ij of the matrix quantify the contribution of pairs of features, I and j, to the distance measure. This lambda ij tells us how important is the combination of feature I and j in the distance function, so also for the classification. And in particular, the diagonal elements summarize the contribution of a single original feature dimension to our distance function. So we can interpret it, at least heuristically, as the relevance of the original feature for the classification. How important is that feature for the classification? Well, one clear statement would be if lambda jj turns out to be 0 for one of the features, then it's not taken into account in the classification scheme at all, and we could tell the domain expert, you don't have to measure that quantity. Our classifier will not use it anyway, right? Now, this direct interpretation assumes implicitly that all features have the same order of magnitude, numerically. Of course, otherwise, lambda jj and the lambda ij will compensate for the different orders of magnitude that you observe in the different dimensions. But that we can achieve easily, very often, by simple things like a z-score transformation that takes care that all features that I'm using in the distance have the same order of magnitude. That's just a technical remark here. Okay, so we optimize the prototypes, we optimize the distance measure at the same time in one and the same training process. And the motivation is, of course, we hope to get improved performance over the simple version, which only moves around prototypes and uses a fixed distance measure, right? We might be able in the end to simplify the classification scheme. We could, for instance, get rid of supposedly non-informative features. If some features are just noisy compared to with respect to our classification, then we can get rid of them, and the method would signal that. Right? We can also use it as for discriminative low-dimensional representation of the data, which is very helpful in interdisciplinary projects, for instance. And in a sense, we could hope that the outcome of the training gives us further insight into the data, into the classification problem, by identifying the most discriminative features, for instance. We might also be able to incorporate, in a way, prior knowledge by structuring omega according to our prior insights into the problem. Right? So all this is possible and can be exploited. But now, I guess, it's time for the practical applications. Let's take this to some data set. So the first problem I want to consider is steroid metabolomics for the detection of malignancy in adrenocortical tumors. Now this is a collaboration, a long-standing, ongoing collaboration with the University of Birmingham, the medical school, and the lead scientist there is Wiebke Alt, endocrinologist. And this was the first important paper in that context. What is the idea? Well, the adrenal gland is a gland that sits on top of the kidney, and it produces, well, in layman's terms, it produces hormones. What it really produces is building blocks for hormones, metabolites, steroid metabolites. But very often, or relatively frequently, doctors find a nodule or tumor in the adrenal gland. Usually this happens incidentally. The CT scan is done for some other reason. There were no symptoms yet, but then once one finds there is a nodule, there's a potential tumor in the adrenal gland. And the question is this, of course, an important question is this a benign tumor or is it a malignant tumor? The malignant variant, the adrenocortical carcinoma, is a very aggressive form of cancer that produces metastasis very rapidly and very aggressively. The benign tumor you can handle, you can treat more conservatively, and it's not as life-threatening. It has effects on your hormone production, it has effects on your well-being, but it's not as dangerous as the ACC. Usually, both cases, there will be a surgery done. That's the standard procedure. But of course, if you would know in advance the tumor is a malignant one, the surgery is usually more aggressive to be on the safe side, right? So this has significant consequences for the patients and for the treatment. Now, the idea of Wiebke Alt was, well, let's look at what the gland does. The gland produces steroid metabolites, and there's a panel of 32 candidate steroid metabolites that one can look at. And what they measure is the excretion over 24 hours of these 32 substances in the urine. And then the idea was, or the hope was, that maybe benign and malignant tumors leave a different fingerprint in this excretion pattern. That was the hypothesis, and if you look here at the raw data, this is color-coded, 32 excretion values for 100 patients with a benign tumor, and here 32 excretion values for roughly 50 patients with a malignant tumor. You see by the naked eye there seem to be some differences, but well, looking at a single column here and say this is ACA or it's ACC, that's different. That's a more difficult task, and that's where we use the machine learning now. Okay, so the aim would be to develop a novel biomarker-based tool for the differential diagnosis of malignant and benign tumors, and the idea is to identify characteristic steroid profiles, in our language, prototypes. So what did we do? We applied generalized matrix learning vector quantization, just as I introduced to you. We, for the classification of ACC versus ACA steroid metabolite profiles, and we do a validation scheme, so we take only 90% of the available data, we keep 10% away for a test set. We determine prototypes, we determine the relevance matrix, which is now a 32 by 32 matrix, easy to handle, not too complicated, and then apply that classifier prototypes together with a distance measure to the test data, and repeat this again and again to get a better idea on average what performance can we expect for new samples for new patients. Okay. So here's the outcome. First of all, we can look at the prototypes, and as I said, this is something that I can take to the medical doctor and say, okay, what I see here is that in the ACC, steroid number 19, THS, is much more expressed than it is in the ACA. Does that make any sense to you? And then they said, yeah, we kind of knew that, okay? But also other things were not so obvious. So this is the level that you can take the discussion to with the medical expert easily. here's the 32 metabolites. You don't have to remember them for the following. I just show them to impress you. Okay, now we also get the relevance matrix. The relevance matrix has off-diagonal elements and it has diagonal elements. And the main message here is the diagonal elements. And it tells us that certain steroid metabolites in our panel seem to be very relevant for the classification scheme, while others are not. Okay, now this could, for instance, be used to suggest a subpanel, a smaller panel of steroids that we want to use in practice. For instance, something like 9 or 10, because there's machinery from the shelf that can measure 10 steroids at a time, which would simplify the procedure, make it faster and cheaper. And in fact, this idea and this realization is patented in the meantime by the University of Birmingham as a practical diagnosis tool. What performance do we get? Well, I'm assuming that everybody is familiar with the ROC characteristics, where you compare true positive and false positive classifications for the different classifiers. If not, please complain. So what we have here is the ROC curves for the plain vanilla learning vector quantization using Euclidean distance. That would be this red curve, right? Then there is a version that uses diagonal relevances in the matrix lambda, and the off-diagonals are set to 0. So this is only weighting the 32 steroids differently, and that already gives an improvement. But for the full matrix scheme, where we have a 32 by 32 lambda matrix, we get an ROC which is much better than the others and has a decent performance in terms of sensitivity and specificity. So we are both. We go here, for instance, we're both able to detect a lot of malignant cases, but we get rather little false alarms only. So this is encouraging and makes a lot of sense. And we see here how the introduction of the more complex distance measure has improved the performance of the classification. Okay, now whenever you work with medical doctors, there's a 3-step strategy that I want to promote. You have to confirm something that they already know first, because then they will be convinced that you're not a complete idiot. You found something that is correct and they knew it already. Next step, you have to surprise them with something that they didn't know yet. And the icing on the cake is you have to visualize complex things in simple graphs, and then they are your friends. So this worked out very well in this problem. Here's the confirm part. Our analysis says steroid number 19 is very important. And Wiebke answered, oh, of course, I knew that. If you look at, for instance, the histogram of steroid 19, THS it's called, for ACA patients and for ACC patients, you see clearly that this is discriminative already to a certain degree. You can do univariate statistics and you will see that steroid number 19 is important. We knew that. Well, we also get from this that steroids like 8 and 12, for instance, are also quite relevant. But on a univariate level, they are not at all. So this is the surprise part. So Wiebke said, but look at steroid number 8. If you draw these histograms for ACA and ACC, it totally mixes. Steroid number 8 has no discriminative power whatsoever. 12 maybe a little bit, but also not very strongly. But our method says 12 and 8 are important, and you can show that by simply looking at the combination. So if you plot here data points, blue for ACA and red for ACC patients in the space of just these two markers, then you see that it's the combination of the two that makes possible a certain discrimination of the two classes, right? And that you wouldn't see in a univariate method because you're projecting to one of the axes only and everything mixes happily. Okay, so this is the surprise part, and here's the visualized part, where we simply take in the end of the training process our matrix lambda, and we do something that is similar in spirit to PCA, but we're not using the covariance matrix, we're using the relevance matrix. So the first eigenvector of lambda and the second eigenvector of lambda can serve as the most discriminative directions in feature space, can be interpreted in that way. And what you see here is the projections of the ACC patients and the ACC prototype. And the blue crosses are the ACA patients, the benign patients, benign tumors, and the prototypes are somewhere hidden here. And you see that this visualizes these two classes very well. And on top of it, I've plotted healthy control patients that were not used at all so far in the analysis. And what you see is that the healthy control happily mix with the ACA. So in this two-dimensional space, at least, benign tumors are closer to control, healthy control, than the malignant tumors. That's reassuring. In other words, if I would see that my control patients spread all over, that would not be a good sign for this classifier, for this method. Okay. So this is, of course, ongoing work. They are replacing the way they measure these steroids by a more efficient one, which is cheaper, faster, but also has some accuracy problems. We have done, in the meantime, a prospective study with about 2,000 patients that we had not seen before. And this study confirms the performance as a classifier very well and is the first step towards actually making it officially a diagnosis tool in practice. Right. We're also looking into monitoring patients after treatment, after surgery, for instance, which has other problems because they are treated with chemicals that mess up the profile inherently. So we have to still find the signal for recurrence in that. We are also applying the same methods, steroid metabolomics and machine learning analysis, to other disorders. The liver, for instance, plays an important role in the metabolics of hormones, and steroid metabolism-related liver diseases can also be diagnosed to a certain extent based on this kind of data and this kind of methodology. Okay. Okay, so second example, same machinery. That's one of the nicest things about machine learning. Once you get your Excel sheet from the medical doctor, for instance, you don't really care anymore what it was. Well, you still should care, but to a certain extent you don't have to. So this is about rheumatoid arthritis, another collaboration with the University of Birmingham, but with the Queen Elizabeth Hospital in that case, Dagmar Scheel-Töllner being in the lead here, and also that has led to a paper in the Annals of Rheumatic Diseases a few years ago. So what is the idea? Very often patients present to a doctor, a general practitioner, with some inflammation in some joints, right? So you might have inflammation here, inflammation here. And the question is, well, some of these patients, if you look back at them in half a year or a year, have developed a serious condition which is called rheumatoid arthritis, and then it's manifest, and it has spread further to other joints, and it really affects your well-being, and it's an unpleasant disease in the long run because it ruins the joints, essentially. Some other patients were the lucky guys where the inflammation resolved because it was not an early rheumatoid arthritis, it was some mechanical effect that caused the inflammation or some other influence that caused it temporarily. And of course it would be highly desirable as early as possible to distinguish between the resolving cases and those that will develop a rheumatoid arthritis after a few months or years, because then you can start the treatment earlier, and obviously that would be a big thing, a big advantage. So that's the ultimate goal, but also one would like to understand what distinguishes the resolving from the early cases. So what are the disease mechanisms that lead to rheumatoid arthritis? And the hypothesis of Dagmar Scheel-Töllner and her group was, well, let's look at cytokines. Cytokines are messenger proteins that the immune system uses, and which get activated in inflammations, and there's a whole zoo of them. And one can measure in the synovial tissue, in the joint tissue, and also the synovial fluid, one can measure the expression of a set of cytokines. And in fact, there were 117 on the panel. They're all cell signal proteins, and they can be measured by taking a sample from the synovium, the tissue in the joint, and then sophisticated techniques to measure their expression. And they are produced by different cells in the immune system, but what you're really measuring is the total expression of these markers. Now, the problem with this data set is, it doesn't say anywhere, that we had on total not even 50 patients in this data set. So the hopes were very low that we would come up with a reliable diagnosis system. But maybe we get, even from this small data set, some information about what are the most important markers in this game. So the goal here is more understanding what is going on rather than coming up with a diagnosis system in the end. That will be rather hopeless. One big problem is, for instance, that you hardly find healthy controls that volunteer for taking tissue from your joint, from the inside of your joints, because it's a painful, risky thing to do. Well, never mind. So we have a feature space which is 117 dimensional, and we have only like 45, if I remember correctly, samples. So that's a problem, of course. So we don't want to use the full feature space. Well, one easy thing to realize without much trouble is to do a pre-processing in which we apply a principal component analysis, easy, standard. And we found that 21 leading principal components explain already a lot of the variation. So we're going from the 117-dimensional space to a 21-dimensional space. That's a more healthy number if you think that if you know that you have only 50 samples, right? Now the nice thing is that we can do generalized matrix relevance learning in that 21-dimensional space, which would look like this. So a distance in this tilde vectors would be of this form with the relevance matrix lambda tilde. But knowing the principal component analysis, knowing the projection, we can write this in terms of the original feature space in retrospect, right? So because it's essentially a linear mapping on top of a linear mapping, and we can then interpret lambda, which is the PC projection transpose lambda tilde psi here, and we can interpret the relevances also in the original 117-dimensional space again. That's fun. So we considered two-class problems. One is should be a piece of cake. That's distinguishing established rheumatoid arthritis from healthy controls, uninflamed controls. And the second would be to distinguish early rheumatoid arthritis from resolving inflammation cases. Okay, so we applied the same procedure. We use the 21-dimensional data. We do a cross-validation scheme, leave two out validation because the data set is so very small. So we leave one out from each class, but that's a detail. And we obtain the receiver operating characteristics. And this is the problem that I said would be a piece of cake. Everyone can distinguish established rheumatoid arthritis from uninflamed control, and so can our system. So this is nearly perfect classification. Whereas for the early cases where there's resolving inflammation, the ROC is better than random. So we're having some signal here, but it's not excitingly good, which you wouldn't expect from such a small data set. The interesting information does not lie in the ROCs. It lies in the relevances mapped back to the 117-dimensional space. And there the surprise was that there were two markers out of these 117 which apparently dominate the game drastically. And the surprise for the rheumatologists was that they didn't have them on the list as candidates, that they would expect to play a big role. Now, this is not as solid as it should be because we have such a small data set and it's prone to discussions and doubts. But of course, it gives you ideas, and it gave ideas to the rheumatologists. So maybe we should look into these markers more deeply, more thoroughly. And then I didn't hear from them for quite a while. But two PhD thesis projects in medicine and a lot of work in the lab later, they said, we confirm your finding. These two markers are important for the disease mechanisms of early rheumatoid arthritis. So these two markers, they come with different names in different communities, but that's the same two markers. They could show by staining techniques based on joint tissue, synovial tissue. They could show that these two markers are very much present in early stages of rheumatoid arthritis. And they could also locate where they are produced. They are produced on macrophages. So we got a lot of insight. They got a lot of insight into the new insight into the disease, into the inflammation processes, and which part of the immune system actually is involved, for instance, based on a rather simple machine learning result, which I found is neat. Of course, as I said, I would never trust this machine learning result as such and say, now this is the tool to diagnose rheumatoid arthritis, but it has given insight that led to further research and further insight. Okay, there's a lot of things that we would like to do in the future. Integrate gene analysis, gene expression data, for instance, from the same patient cohort, but all this is very difficult and long-term on the medical side to realize, obviously. Okay, third example. And from example to example, the data gets higher dimensional. I started off with 32 steroid metabolites, then 117 cytokines, and now it will be 300,000 voxels in a brain image. Sounds scary. So this is together with people from the Department of Neurology in Groningen, the Neuroimaging Center, and several centers in Spain and Italy are involved in gathering the data. So the ultimate goal, we're not there, I'm not claiming that, would be an early diagnosis of neurodegenerative disorders like Alzheimer's disease, Parkinson's disease, and so forth, based on brain imaging. And the technique that's used here is FDG PET scans. So this essentially measures the glucose uptake in the regions of the brain, and that is used for the imaging. So the data is acquired at 3 different medical centers who told us, well, we're using the identical equipment, we're using the exact same pipeline, we're using the same pre-processing. This data is homogeneous, you can use it as one data set. No, we found out later that that was not quite true, and I will show you how. The analysis we do is by machine learning, and we use, of course, GMLVQ, because I'm talking about this mostly. Also in the localized version, we're using the support vector machine for comparison, and we use data from different centers, across centers, and so forth. So the question is, can we come up with a reliable FDG PET-based diagnosis? Is our classifier, if we train it on some data set, is it compatible across different medical centers? So can we ultimately obtain a robust universal classifier that could be used in all of these centers? Now, FDG PET scan images are based on, as I said, glucose, which is marked radioactively. You get images of this type. If you want to see more, you can go to the webpage of this GLIMPSE project. And we focused on healthy controls, patients with Parkinson's disease and patients with Alzheimer's disease. Number of patients is reasonable, but still not big. That's a genuine problem, or a frequent problem in medical data, of course. Comes from 3 centers, one in Spain, one in Italy, Genoa, and one from Groningen, and we have different numbers for the different conditions. Alzheimer's was not present in the Spanish data set. Okay. So the idea is the workflow. We have, did I say 300,000 voxels? It's only 200,000 voxels, apparently, per patient in our 3D image. And then there is a standardized methodology within neurology to project this data down to a low-dimensional space. And essentially what is done there is a PCA-based method together with some other tricks of the trade, which is performed with respect to a reference group of subjects. So we used the same reference group of subjects for the PCA. They used it. But it's separate from the actual patients that we look at. Okay. Details of the processing can be found in the literature. What you get from there is subject scores. Per patient, instead of the 200,000 voxels that you don't want to handle, you get on the order of a few 10 patient scores, which you use as your feature vectors. And then we of course know the condition of the patients. We know the condition of the subjects in this space, obviously, and now we can train some classifier and apply it to some test data, and that classifier should then assign every test data point to one of the 3 conditions that we're interested in. So the scheme is the same as before in the other problems, essentially. So we used a support vector machine with linear kernel. That's the term that you have to use for a few years now. The elder ones among us remember that this is just a perceptron of optimal stability, but okay, never mind. So it's a linear threshold classifier that realizes a large margin between the 2 classes, and we allow for certain errors in practice here, because we know that the classes will not perfectly separate in this simple scheme. Never mind. Second classifier we use is generalized matrix learning vector quantization, and the third one is the localized version where each prototype has its own relevance matrix. Okay. Then we perform 10-fold cross-validation, 10 randomized runs of 10-fold cross-validation. We get accuracy, sensitivity, specificity, receiver operating characteristics. And in earlier versions of this work, we tried also decision trees, but they were outperformed by all these methods that I'm using here. What are the main results? And we're approaching the end already, so relax. Well, we get relatively good performance of the classifier if we look at data within the same center. So here we have only data from San Marino in Italy, and we can, for instance, discriminate very well Parkinson's disease versus healthy control, Alzheimer's disease versus healthy control, and also Parkinson's disease versus Alzheimer. And similar, we see very good performance also if we formulate this as a 3-class problem right away, but this is nicer to look at. So we get area under the curve for the ROC, which is 0.8 or sometimes even close to 1, depending on the difficulty of the individual problem. So that's good. Within the center, we get reasonable performance. Is the performance compatible between centers? So what happens if we train our system on data from the Spanish center, Navarra, and test it on San Marino data? Not so good. It's still better than random, apparently. Here we train on San Marino, test on Groningen data. Here we train on Groningen, test on Navarra data. It's still okay-ish, better than random, apparently, but the performance has deteriorated a lot compared to the within-center performance. So there's something going on. And my suggestion then was that the neurologists didn't like it all. Let's check whether we can train a classifier to distinguish the centers from the data, which would contradict their claim that the data is essentially homogeneous. So can we infer the medical data center that produced the data from the data? And here's the area under the curve of the ROC. So we can, this is an example where we distinguish Navarra data from San Marino data flawlessly, right? So it's perfectly distinguishable where the data comes from. So there's something wrong in this assumption that the data is totally homogeneous. What are the possible explanations for that? This is ongoing work, so now we're in heavy discussions with the neurologists, as you can imagine. Maybe the preprocessing is not quite the same. Maybe they're just using different versions of some software. Who knows? We have to check this out in great detail now, right? But maybe also it's different patient cohorts. Well, in case of the healthy controls, we can assume that healthy controls are pretty much the same in San Marino as they are in Navarra. But maybe the threshold for sending a Parkinson's disease patient to the facility is higher in Spain than it is in Italy, something like that. So maybe they are looking at more severe cases than they are in Italy. And that would explain, could partly explain this discrepancy, right? Okay. The nice thing, the positive thing about this is that the method allows us to check for this, right? So this here's a nice visualization that I come back to my third part of convincing medical collaborators, visualize the data. What you see here is a projection on the first eigenvector of the relevance matrix and on the second eigenvector of the relevance matrix for the 6-class problem where we have healthy control and Parkinson's in the 3 different centers. And you see, well, left of the red line and right of the red line, that discriminates healthy controls and Parkinson's disease patients. But here I have these 2 blobs here are all data from Genoa. These 2 blobs are all data from San Marino. This is all the data from Groningen. So in this visualization, you already see that there's one direction in the classifier, in the multi-class classifier, that kind of distinguishes the centers, and the other one distinguishes the condition of the patient. So what we can now look into in detail into what are the features that distinguish the diseases and which are the features that distinguish the centers. But we're at the beginning of this exercise. We can even go back to voxel space and show images to the neurologists. We can take the prototypes and project them back roughly to the image space and then show them slices of the images and ask them, do you recognize some patterns that you would have used in your individual diagnosis, for instance? We can also look at differences between prototypes, compare Parkinson's disease and Alzheimer's disease, and then tell the neurologist apparently the largest differences occur in this and that area of the brain. And then they can tell us that's nonsense, or well, maybe we have to look into that, right? We can also look at the eigenvector of the relevance matrix. This would be the direction that distinguishes the conditions of the patients, and tell the neurologist, apparently it's very important to look at this part of the slice number so-and-so in the brain scan. Okay. There's a lot to do in this part, so this is an ongoing project, obviously, and I'm only showing you the first results. But what I want to motivate and make clear is that the method allows you to interact closely with the medical experts and give them valid information and have a chance to discuss things with them, not just present a classifier with a certain performance and that's the end of the story. Okay. Summary and conclusion of my talk. I hope I have convinced you to a certain degree that prototype and distance-based systems are very useful frameworks for classification, and they can also be modified for regression. They're intuitive, transparent, and interpretable, and they're easy to implement, actually, very flexible, natural for multi-class problems. I discussed only classification, and mainly that's what we have done with them, but you can use them in different contexts as well. The addition of relevance learning makes them more powerful and even more flexible, and it allows for further insights into the data and into the problem that you're attacking. So all these properties make them very useful in biomedical problems. There are a couple of articles that I can advertise here. So we have written a review on general prototype-based models in machine learning in the Wire's Cognitive Science journal, and then there's a review that was actually an invited lecture at a conference on biomedical applications in the conference on algorithms for computational biology. Okay, now if you're interested in trying out the method, there's lots of implementation now available in the web. There's our own, this is my own, I call it the no-nonsense beginner's version of GMLVQ, the simplest version. Kerstin Bunte has implemented also several other extensions, including the local relevance matrix. For the Python people, there's a scikit-compatible collection of code from our friends in Bielefeld, and there's even a Java plugin for Weka learning environment available in Mitvida. If you go to my homepage, my personal homepage, you will find all these links. This is the homepage. Well, I won't go through all these challenges. I want to conclude with something that kind of connects to the previous talk. It's in applications, it's always important to incorporate knowledge from the domain and talk to the domain experts, include it in what you do, make use of what is known. So this relates to the previous statements made in the previous talk. And this is a nice illustration that I found recently on Twitter, actually. Of course, cutting the tennis balls in half lets you store 2 more balls in the same space, but had you talked to a domain expert, i.e. a tennis player, he would have told you that it's not a good idea to do that. So keep in mind that it's always important to talk to the experts if you want to do practical applications. And exchange information and make use of everything that's available. And with that, I would like to conclude and thank you for your patience.
I noticed that you have tried to project the data on a relatively lower dimensional space. Is it because the number of data points is not large enough? Or is it because of computational constraints to run really the algorithm?
This relates to the statement that I made vaguely earlier, that the relevance matrix in generic cases becomes low rank in the training process. So depending on the structure, in a 2-class problem, typically in the end we see that it's dominated by one eigendirection only, typically. In a multi-class problem, it's several. But it goes down to very low rank, and we can understand that mathematically from how the training is done. So we can show that generically the rank of the matrix will reduce in the course of training. And that's, and fortunately that is so, because otherwise you would definitely expect overfitting, right? The thing is, you're guiding that low dimension, you're finding that low dimensional space, and the thing is guided by the discriminative nature of the task. So you're looking not just for some low-dimensional representation as you are in PCA, where you only take variance into account. This is guided by the performance in terms of the classifier. So you're finding a good low-dimensional representation. But in detail, it depends on the data set. If it's a very difficult data set, then this two-dimensional projection will not show you anything. Maybe you need an 8-dimensional projection to do the classification properly. But the system will find out. That's the nice thing. Sorry.
I have the micro. Yeah, thanks for the talk. I have one question. You had in your examples typically quite cases where the two classes or the three classes were more or less of the same size. In many problems, you have something where you have one class which is very small, I don't know, some positive, and all the rest or so. Is this feasible, or is this a problem?
No, that's a problem that's common to most machine learning analysis of real-world data, obviously. So there are techniques, undersampling, oversampling are the keywords, where you try to, at least for your training data, realize a balanced set of data. Now, you cannot cure the problem totally, not completely, but for instance, the ROC analysis, the receiver operating curves, already gives you some information of what the influence of the imbalance would be. So maybe you get from a training on an imbalanced data set, you get a classifier that, let me go to the ROC first. Where's my first ROC? Just a second. Yeah, maybe here. Maybe your training data is so imbalanced that the nearest prototype classifier would sit here, right? But then the ROC tells you, well, bias your classifier in the right direction, then you can go along the ROC, you can go here for unknown balance in the data, in the working phase, right? So it's an intrinsic problem. In medicine, fortunately, it is always there, because usually you would hope to have much more healthy control data than you would have for a particular rare disease or something. But yeah, there's ways to handle it, but they're not perfect, obviously. But this is not a problem of this method. It's of any machine learning analysis of medical data or other real-world data. You have that kind of problem. Yeah.
So I have more a technical problem on the case of the neurodegenerative diseases.
Mm-hmm.
For your inputs, what did you use actually? Did you use the direct slices like these from PT, or did you use the 3D tomography files actually?
Yeah, this is just for illustration. Well, that's the part that the neurologists do for us. So they take the 200,000 voxels, sorry, and they have means to mask them, to register between different patients, and then project down by this scaled sub-profile model PCA-based method to a low-dimensional space. And that's where we come in.
Okay, so you don't start from the 3D raw files?
We do not. No, the neurologists do. Yeah. Yeah. I mean, there were suggestions from the neurologists, well, everybody's doing deep learning now, why don't you use a deep neural network on this 200,000 voxels? And they said, okay, then bring 1 million examples, then we will do it. But that ended the discussion then.